Well, good afternoon, everybody. My name is Bob Wright with the University of Nebraska, and Tom Hunt, also with the University of Nebraska, and I will be the moderators for today. As we get started, we wanted to thank several people make several thank yous, particularly Phyllis Bongard with University of Minnesota Extension has done all the local arrangements to set up the website and this Zoom meeting, and a lot of other things. She's working right now to make sure everything flows very well. We're going to be talking about a lot of different research wouldn't have been possible without funding from several groups, in particular the North Central Research Program, which gets funding from state commodity boards, several state soybean boards, and several USDA programs, North Central IPM Center, USDA NEFA grants, several industry sponsors, and other state funding. And we also want to thank a lot of the farmers. A lot of the research we're going to report today is based on-farm research in commercial fields, and we wouldn't have made as much progress as we have if it weren't for all the cooperating farmers we've worked with. So I'll turn it over to our first speaker here, Justin McMecken with the University of Nebraska.
Thank you, Bob, for the introduction. And I'm going to kick things off today by showing you how to navigate our website, soybeangallmage.org. Just a couple kind of key things to look for on that website. Then I'm going to hit some field surveys that include both new detections and how we're doing that type of work, as well as our historical counties where we have infestations in them or states and what we're finding in those states. I know a couple of you in the pre-question asked, is gallmage getting worse? What's happening over the years? And then we're going to take a quick kind of crash course in biology to give you a bit of a refresher if you haven't seen the other webinars. And for those of you that are new, hopefully enough to navigate the presentations as we go through them. So I'm Justin McMecken. I'm a crop protection and cropping systems specialist. The other kind of key people in other states that are working with us are listed on here as well. And as an intro to soybean gallmage, if you've never heard of it and you just jumped onto this webinar, it's kind of classic orange larvae here at the base of soybean plants, the adults, and then under kind of our worst case scenarios, the amount of injury that we can see in the field. So starting with the website, this is the website. When you first come into the web page on the left there, you will see, of course, emergence scouting and management. That orange box that's partway down is kind of your first place to look for any updates on the web page, things that might be important to you. Typically, we list the date there during the season. There are links and important information about emergence of soybean gallmage and other things related to its distribution. On the right-hand side is a bunch of buttons that are just below that initial listing. And I'll just go through these each just to know what is behind each of those buttons if you click on them. The first is the distribution of soybean gallmage. You could see this in a number of talks that we've given in the past. The color coding is the years at which soybean gallmage larvae are found in those counties. And you can see that in 2022, we are still finding new counties for soybean gallmage. Less so than we did the previous year in 2021. This does not indicate spread of soybean gallmage. We're getting better at detecting this insect, so we're not going to count those as spread, at least initially. Scouting is another resource tab for you. Here's just 2 videos that are available. You can watch these videos. It provides you how to scout for soybean gallmage. If you're still wondering how to scout or need more information, there are a good 6 hours of recordings under this Midwest Soybean Gallmage series recordings that are from our previous year's events that have some nice scouting information that Aaron Hodgins presented and others. Notifications is another tab that's on here. The notifications are updated during the season on key events. And so if you're on our alert network, which I'll talk about in a second, when you jump onto that, that's the first place to go, where you'll see kind of detailed information on where emergence might be occurring, some management recommendations, or the presence of wilting or dead plants in the field. Another important resource is to contact us. If you're in one of the 4 states where it's been found so far, or 5 states, minus Missouri, which we have a new person starting in Missouri. This is where you can find kind of key people within the state to contact if you have questions about soybean gallmage. And then many of you that are probably on here are already on this alert network, but if you want to join to receive an automated text message, phone call, and email, or at least one of those, you can sign up by clicking on that Join the Alert Network. It requires your name and first name and last name and an email at least. You're welcome to add your phone number. And then in the additional comments, if you only want to text or just a phone call, then you can indicate in that area. Just below all of that information are maps that will appear during the season. First and initially, you'll just see this map with the yellow text on it. This is when the gallmage is emerging from last year's soybeans, which we'll touch on here during the talk in detail. But this is when we first see soybean gallmage emerging during the season. The maps are the same. So whether it's the overwintering generation or once they start emerging from the current year's soybeans, the dots represent the same type of information. If it's a black dot without a yellow center in it, it means we have a trap site, but we haven't detected any emergence from that site yet. Once a site turns yellow with this little yellow center, it means that we've had activity at the site, and the number below it, yellow or green, means the number that we've captured so far that season. And just below that is a plus or a plus 0 or plus some number above that that indicates the amount of activity since the last sample. And last samples vary between states because of the distance to travel to the sites. They're anywhere from a day to 2 days up to a week or so between each one of those collections. And then underneath each one of these is the last update on when that update occurred for that map. So I want to move and spend the rest of the talk here kind of focusing on field surveys and then that crash course in biology. I just want you to know that we do field surveys each year in adjacent or adjoining states where we haven't detected soybean gallmage or its presence has been fairly limited in the case of Missouri. And we provide those individuals, they scout well beyond that. I know Janet's on, she does a lot of scouting beyond that in North Dakota. But scouting along these field edges, we provide, or these state edges, we provide information on USDA Cropscape, which tells us the corn and soybean histories in those areas, increasing the likelihood we have corn or soybeans next to one another each season. And then we also use dense vegetation maps in order to determine high-risk areas to scout, increasing the likelihood we might detect soybean gallmage when it's in low number. And that hopefully, explains to you why we've been finding them in lower numbers in some of our states and why those maps have expanded. For anybody who's scouting for it in areas where you haven't seen it yet, wait between R3, which is beginning pod, to that later, seed development stage, R6 in soybeans. Just catch them before they are going to begin to mature. Okay, I'm going to lean in and focus on some more detailed information, first in Nebraska, and then I'll show some information on Minnesota as well. We take a lot of data within the state. This is courtesy of Nebraska Soybean Board funding. You'll see their logo here over the next couple slides. We collect a lot of detailed information within each site. Tom Hunt and I do this in Nebraska. Bruce Potter does this in Minnesota, and Aaron and Adam do this in Iowa and South Dakota for detections of presence in the state. So a number of you asked, what has happened with soybean gallmage over the last few years? And there are 3 slides and 6 maps for Nebraska that highlight kind of what we've seen since we first started doing these surveys. I'll have you focus on the map that has larvae per plant first. You can see that in 2020, when things were pretty bad, we were finding a lot of averages on the county level for the fields surveyed within that county of greater than 20 larvae per plant. And so you can see the number of dark and red counties that are in there, some lower, all in eastern Nebraska here for detections. The wilting or dead plants really correspond to where we find large larval numbers, not always, but in general, they correspond to where more plants are present. This is also along the field edge within the first 50 feet for an average of wilting or dead plants. In 2021, I'll just flip back and forth here so you can see a reduction in the number of counties that have greater than 20 larvae per plant. But you notice we still have quite a few. And of course, we've increased the distribution of soybean gallmage where we're detecting it in the state. Good news for farmers, a lot of that wilting and dead plants went down, but we're still prevalent in a number of counties in eastern Nebraska. No new detections where we were finding soybean gallmage you could see have any wilting or dead plants. If we look at just this past year, a pretty similar distribution and abundance to what we saw in 2021. Soybean gallmage is out there. It exceeds the 20 larvae per plant average in some counties, but far less than 2020. and that it's obviously present out east. We have a few counties that have popped up further west that do have some wilting or dead plants, but very low frequencies, at 0.1 to 3%. So nothing to be concerned about. This is Bruce Potter's data from Minnesota. They've taken, as you can see, these are individual points in the field or across the state where they've been, looking for soybean gallmage. The green or gray dots indicate there is an absence of soybean gallmage and no symptoms in the field. Red or yellow indicate the presence of symptoms in the case of yellow, or red symptoms in soybean gallmage found. So you could see a lot less pressure in Minnesota than what we see in Nebraska, but soybean gallmage presence in the state increased between 2020 and 2021, perhaps because of detection techniques or other things. A lot more research needs to be done in that category. So this is the website will keep you connected. The distribution maps, scouting, and adult emergence maps are there. In-season notifications on adult emergence. Those surveys show that soybean gallmage is frequently found in soybean fields in Nebraska. About 77% of historical counties last year had presence, less so in Minnesota. And plant injury varies year to year, but typically corresponds with a lowerable number. I'm going to spend just 2 or 3 minutes here giving you a crash course in the biology of soybean gallmage. It'll focus around this life cycle you see here. Hopefully this is a refresher for many of you, and if not, it's enough that you can move forward with the rest of the talks and gain some information. So I'm going to start with silken cocoons. They're present from last year's soybean field where we see injury in the field or presence of larvae in the field. The larvae fall off the soybean plants overwinter in the soil. Mika De Silva Lima will talk here in a little bit and provide you a lot of detail on this part of the process. The adults emerge in early spring, around late May and early June for each year. For the last couple of years, we'll talk about emergence more. And then they infest the closest soybean fields that are in the area, or typically we see pretty heavy distributions next to those fields. The females have a long ovipositor. They're indicated to only live for a few days, but they seek out plants that at least have fissures or cracks near the base of the plant that form around the V2 stage. V1 stage plants with a single trifoliate don't have any cracks. Once they get in and start feeding in those cracks, they cause these dark discolorations along the area where the fissures are present near the soil surface. This is the cotyledonary nodes and soil surface. And that dark discoloration is, if you peel that area back, you'll find multiple different larval or instars of soybean gallmage present, as you can see in the photo here, and all can be found throughout the course of the season after those initial infestations occur, at least in Nebraska. And it's really hard to know the number of generations, but we expect there could be roughly 2 generations turnover in the current year's soybeans. So take-home message, soybean gallmage overwinters as third instar larvae in silken cocoons. At least that's the indication from the data we have so far. They emerge from last year's soybean in early June. The fissures are present at the base of the stem. They're critical for infestation. All 3 instars can be found in a single stem. And we likely have 2 generations per year, although not always the case or easy to see, especially in our northern states. So with that, I will stop and take any questions if we have time. I know I went a little longer covering that biology.
Can you hear me, Justin?
Yes.
Okay, we don't have any new questions, but we do have a couple questions you might quickly address from last time with respect to distribution. One is about how far, and this is more for Aaron, about how far east into Iowa have we seen the gallmage?
Yeah, great question. It covers basically the eastern half of the state. Approximately 41 counties are infested, so it's not quite in Story County. If you ever asked the question, it's not quite there, but it's basically all the counties just on the western half. I think maybe I said the eastern. It's the western half of Iowa.
Okay, and another question is, this is kind of a general question, and you mentioned those of us that are working directly with it, but you have contacted a lot of other entomologists in the north central region particularly. And this question is basically, do you expect it to spread nationwide, or at least in the soybean growing region of the US? And what are your thoughts there?
Great question. I think everybody's wondering that question. I don't think anybody has the answer to that, knows. and others feel free to chime in, but we're obviously aggressively trying to find it in other states. The climate in theory matches that, but we've not seen it rapidly spread to those areas. So I think it's time spent working with soybean gallmage and then continuing to track for it. I don't know if it'll ever go nationwide, but certainly we're kind of seeing this, we're finding it in new areas, but in low numbers.
Okay.
Others want to comment on that?
Yep, and I think a key point is that there are other entomologists that we regularly interact with in particularly the North Central region who are aware of it, look for it. And then of course, people like, Aaron and Bruce and Bob and are checking on the edges of their states where it's less prevalent. So I think Justin's right. I wouldn't expect it to spread everywhere. But we do have a network of people that are actively looking for it to see if it does spread. And then another question. Do we have time for another question? I'll just give it. Here's one thing is we noticed in your maps that worse one year and then it started to slack off a little bit and then kind of was the same the next year, which was last year. So is it becoming more of a problem, staying the same? Or is it just kind of fluctuating at kind of a stable state? And I know that's a hard question given it only was identified a few years ago, but what are your thoughts there about the state of its problem causing?
Yeah, it depends. It depends on the grower. I've worked with growers that have had a problem every year since soybean gallmage was identified in 2019, and others have seen it reduced and disappear out of their system. So I think it depends. Why it depends, I don't know. I think there's environmental factors at play, in my opinion. Tom, you and I are involved in a detailed survey in Nebraska here to try and identify risk factors. Moisture and other things are probably part of that based on some research on adult emergence and artificial rainfall. But there's just a lot of things, like you said, we don't know because we don't have enough time.
Okay, and just one final question, which is more of a speculative one, and we talk about this ourselves, is it just chance that the midge began where it has? I mean, why here? What do you think, what's gone into that?
Yeah, man, this could be opened up to all the entomologists to respond. I mean, we've all discussed this at great length. Why here? Why now? We haven't identified a native host yet. To at least this area. Bruce Potter's going to talk about hosts here in a few minutes. But it could have come from some native host in the area. It certainly popped up over a very wide area in a short period of time, suggesting it was kind of seeded into the system. Perhaps our earlier planting dates. I mean, there's a lot of potential, ideas on how it got into our system. But others want to comment on. We don't know the origin. I guess to answer the question, we don't know where it came from.
Yep. I think I see one final question. Well, there's some that pop up as we go along, but one is just a technical one. Do we have a good photo of the silken pupil case? And I'd say yes. You have a variety of photos, and so if you want to see those, looking at some of the presentations that are on the soybeanmidge.org website, you could see those because I believe there's a few there, or I think we'll probably see some as we look through this webinar today.
Yeah, just have nice photos.
So I guess that's for the questions. I guess we can move on for the next talk now.
Okay, great. Thank you, Justin. Our next speaker is Natasha Umezu, is a student at UNL, and she'll be talking about some of her research on soybean gallmage. So Natasha.
Okay, good. So, well, thank you for the introduction, Dr. Wright. And so as Dr. Wright mentioned, I'm a master's student at Dr. Justin McMicken's lab, and I will be presenting some results from our study about the hail damage and the interaction with soybean gallmage infestation. So before I start to show some results, first I would like to explain the reason that led us to conduct this study. And as Dr. Justin McMeekan mentioned in the previous presentation, we can find soybean larvae in the base of the plant because it's the area where we can have this natural fissure in the plant, as you can see here in the first picture. And these fissures can work as an entrance where the soybean gummy adult can start to infest the plant, right? So when hail occurs, we can have those type of damage along all the plant, as you can see here in this second picture. And we believe that this damage could also work as alternative entrance for soybean gummy to infest the plant. So because of this, we decided to evaluate if the hail storms could impact in the soybean gummy abundance. So for this study, we used this machine that was developed at the University of Nebraska, and we used the machine to simulate hail in our plots. And since the time today will be short, I will jump already for the results. But if someone has any question or want to know more about how the study was conducted, please contact us later, and then we will be glad to answer any question about this. So starting with the results from 2021, in the first graph, I have the average of larval number per plant and per treatment. And as you can see here, when we had hail, we could have more number of larvae per plant. So it means that the abundance of soybean gummy was higher. And in this study, we also evaluated the abundance of soybean gummy per hail damage score, because we would like to understand if, depending on the severity of hailstorm, if this could also impact in the soybean gummy abundance. So in the second graph, I have the average larval number per hail damage point. And here in the fissure is the natural infestation, since is the place where the soybean gummy adult starts to infest the plant when we don't have hail. And each number here in the graph represents one hail damage score. So starting with number 1 is when we had a lighter injury caused by hail, and it will increase according to the severity of the damage. And with this graph, you can see when we had a more severity damage level, we could have more larval number. In 2022, we also conducted this study, but in this time, instead of understand the impact of hail, we also would like to understand if the timing that the hail occurs, if this could also impact in the soybean gummy abundance. And for this year, we didn't see any significant difference between the timing that the hail occurs and the no hail. And then also with this study, we also evaluated the abundance of larval of soybean gummy per hail damage score. And again, in the second graph, we can see that when the damage was higher, that was more severity, the hailstorm, we could also find more larvae per plant. So this year in 2023, we will conduct the study again to confirm the data that we already have. But even if, since we could have different data from different years, but what we could see with this study is that the injury that caused by hail could increase the number of larvae per plant. So it means that we can have a higher abundance of soybean gummy because the insect can have a more alternative to infest the plant. But even if the hailstorm occurs, it will depend the severity of the storm, because as we could see, when we had a heavier damage, we could have more larvae found. So this will be important to be considered for growers when they have hail in their field. So it's important to consider to scout the field to evaluate the soybean gall midge injury, since it looks like it's an important risk factor for soybean gall midge infestation. So this is what I have for today. Thank you. And if you have any other questions, please contact us by email or for the website.
Okay, I think we wanted to move on to Bruce Potter. We'll come back at the end of these two talks and try to respond to questions. But Bruce, are you ready to speak?
I'm working on it.
Okay.
You guys see my screen okay now?
Yep.
Okay. Well, thanks, Bob. I'm going to talk briefly about a project we've been working on for a couple of years here, trying to identify some alternate hosts for soybean gall midge here in Minnesota. And this map Justin showed earlier is kind of where we've been finding the red dots illustrate where soybean gall midge has been found in Minnesota. Previously, it's been found on larvae, been found on soybean, of course, sweet clover, and to a much lesser extent, alfalfa. Minnesota has a significant amount of dry bean production, and there's an area of the state where dry bean production and soybean gall midge overlap. And the question I had was, since this is a new pest, we don't know much about it. Can soybean gall midge infest legume crops other than soybeans? So to answer this, we decided to take a different approach. We went with a mobile sentinel plot approach. And what we did was grew different types of legumes in pots, took measurements on growth and the development of stem fissures. And then we put those plants out into soybean fields when overwintering adults and first generation adults in soybeans should have been active. This graph is showing the different types of plants we've tested. I'm just going to show you the interesting bit, and that was that first generation in 2022. We've had 3 varieties of soybeans, and the bars represent the proportion of stems that were infested with gall midge. No surprise, soybeans, gall midge like soybeans. Most of these plants are hypogeal, or in other words, the cotyledons come above ground when they germinate. But we had some epigeal varieties in there. We're throwing a lot of stuff at the wall to see what would stick. And the interesting thing is, we did get some hits on Phaseolus or the bean family. Phaseolus vulgaris is where we have the dry beans and snap beans, that sort of thing, and also lima beans. So looks like soybean gall midge could infest things other than just soybeans. Similar to the portion plants infested, soybean sentinels had a higher level of larval presence or larval infestation. If you look at the lima bean and bean, those infestations were much less, and not all of them were infested. We have a few that we didn't pick any larvae up. So to back this up, in that area that was shaded on the map, we did a survey, and we did find 2 navy bean fields in Lac-qui-Parle County that were infested. Similar to soybeans, the field edges next to last year's soybeans, everything was similar. There was a tendency not only to find them under the outer layer of the stem, but also it looks like some of those infestations started at where branches or leaf petioles attached to the main stem. These larvae were identified by Bob Cook's lab, both by morphology and also DNA. So here too, it looks like the infestation rates on bean were much less than they were on soybean, but the proportions or ratio was real similar to what we found in the sentinel plots. I mentioned we took some growth measurements, and the bars that are shaded did have soybean gall midge on them, or in them, in the sentinels. And here we've got a portion of the stems that had those growth fissures that were mentioned earlier. Soybeans are much more aggressive at producing those. And we've got a few varieties, for example, pinto bean, tiger eye, that were infested but did not produce fissures the same way that soybeans did. Soybean fissures are pretty darn aggressive. High proportion of the stems have those, like I mentioned, and they're pretty deep into the plant, pretty accessible to the adults. Bean, on the other hand, is a little bit different. We've got a plant on the left. That's actually a pretty rare fissure. A lot of them were more superficial and real shallow, just the outer layer of epidermis, and then they suberized over real quickly in the greenhouse. And then a little final piece of information is when we looked at stem diameter, if we looked amongst the Phaseolus plant cultivars and species, there's definitely a tendency for the ones that were infested, the shaded bars, to have larger stem diameter. I don't know if the adults are preferring those, or that has something to do with larval development. And this is 1 year's data, but it's definitely, I think, worth exploring. So I'm going to summarize this real quick. And we do have two new host species, bean and lima bean. And the other plants that I mentioned, sweet clover, alfalfa, soybeans, those are all Asian and Eurasian in origin. And now we threw a monkey wrench into this as far as where this insect is from. Bean is Central or South America in origin. And it looks that glycine max or soybeans is a better host than dry beans. But I still think that dry bean growers, especially in those areas where soybean gall midge is present, they should be paying attention to any kind of wilting or brittle stems on the edges of their field. So that's it. If we got time for questions, I'll try to address them.
Okay, Tom, let's try to address questions to either Natasha or Bruce.
Yeah. Well, there was a question related to hail, and I think Natasha's typing an answer to this, but how long after hail damage should one start to look for infestation?
Okay, well, thank you for the questions. So for this study, we collected our samples after 14 days that the hail was applied. But before this, we already could see some larvae. So if I'm not wrong, it was about 5 days after we hailed. So between those timing, you can already start to find some larvae. But it's important to mention that it will depend when the hail will occur, because if it's in the beginning of the season, so maybe the infestation can be a little bit lower, since the overwintering generation will be present in the field. And then if it will be later season, you can start to see larvae earlier.
Okay, thank you. Another question has with respect to oviposition, and that was, does there need to be some type of opening or wound, natural or otherwise, on the stem for the eggs to be laid? And so far, on soybeans, that appears to be what is necessary, some kind of opening. But again, Bruce threw a monkey wrench in there. It looked like to me that on the dry beans, you saw some oviposition at petiole locations. So there's still some things to look at there. But so far in soybeans, by far the majority of infestations and everything I've seen, it's been at some type of opening, whether it be a hail injury or fissures. It remains to be seen what other types of injury might allow oviposition. We're looking at disease and other things also. So far, yes, except for a couple things that have been seen on Phaseolus. And maybe somebody else wants to add to that.
Yeah, well, Tom, I don't, those bean plants, there might have been injuries on those as well. It's just that they weren't
Okay.
developing the fissures like a soybean would, at least at the same rate.
Okay, sounds good. Now, there are a couple more. Do we have time for a couple more questions?
I think
guess
so.
so.
Yeah.
Okay, there were a couple questions basically on the hail study. And just to get your opinion, why, it seemed like in year one of the simulation study, you did get a lot of infestation on the simulated hail plots. But on the second year, even though you got some, you didn't get nearly as many. And do you have an idea why that might have been? Just a density of midges around, or what do you think happened there?
Yeah, this is a good question. So between those years, we had difference in the pressure, I can say, because 1 year was drier, and then the emergence, we had some difference. But if Justin want to say something, but I believe one of the reasons is because in the first year we didn't have as many hail during the year. Because we simulated just once, and then in the second year we simulated in different timing. So I believe this could also affect the difference between the results that we got between the years.
Yeah, Natasha, I think you did a great job. It's a confusing study, and that's why we presented both years to kind of throw a wrench into people's understanding of that. In part, we're looking at every damage point. So if there's an absence of gallmage, that draws down those averages. When you have less hail plots and severe late hail, it seemed like things were pretty well infested. So maybe we timed adult emergence well with that. But who knows? I mean, that's why you're doing it in 2023.
Okay. And then Justin, there was one final comment more than a question from Janet Knodel. It was and there was also a question referencing this about how far north have we seen this midge go? And so would you like to address that? She had seen a tentative ID, but where do we stand there?
Yeah, thanks, Janet, for that comment. And I think we're all fairly convinced that's soybean gallmage. We just couldn't get a really good genetic match because of the sample. But yeah, Janet has done a tremendous amount of work. That I think in adjacent to in Minnesota with Bruce's stuff is the furthest north we've seen it on the map.
Okay, so possibly in North Dakota, and certainly on Bruce's map, we saw him go ways up there. Yeah, at low levels. Okay, those are the questions I have to date, or to this point. So, any comments?
Let's move on to the next speaker. Going to change topics. Another graduate student from UNL, Mika Ellison, is going to talk about his work with overwintering soybean gall midges.
All right. Thank you very much, Dr. McMecken, for the help with the slide, and thank you, Bob, for the introduction, Dr. Wright. So hi, everybody, welcome. And yeah, I'm a student here. I'm a master's student. I'm advisor of Dr. Just McMecken. And I've been working with soil samples and with the distribution of soybean gall midge in the soil. And today we'll talk a little bit about some of the results that we've gotten so far. So, oh yeah, Justin, you can go ahead, sorry. Yeah, this is a busy slide, but thanks Justin for introducing recall the soybean gall midge life cycle again. Here, what I want to call your attention is just to the end of this feeding period here of the larval stage. When we go and get the end of the feeding period, you can go ahead, Justin. We have that moment where the larvae jump in the soil and then go to this next step under the soil. And we've been seeing so far that track adult emergence is pretty complicated just because we have adult emergence and activity along of the whole growing season. So a couple of studies have been developing to understand this soil stage just because it's easier to track. Go ahead, Justin. Yeah, so with that, a couple of my objectives here is to understand how is the abundance of soybean gall midge in the soil from infested fields. And today, our main focus here will be to recall a little bit from last year there is the presence of gall-mead in the soil profile. And the second one will be to see the abundance along of the growing season. So to understand a little bit of those objectives, we performed some field studies in the years of 2019, 2021, and 2022 in those counties and locations here in Eastern Nebraska. And for the soil depth study, this is something that we shared last year, but just to recall, we collect a couple of soil samples without to deform those soil samples to see how was the abundance of soybean gall-mead cocoons and larvae along of the soil profile. We cut those samples in layers of a couple of inches, 2 centimeters. It'll be hard to convert this right now. And then we process those samples to see how was the abundance in each of those layers. You can go ahead. And here we have the results from 2019, 2021, and the new results from 2022. Those are the result of the distribution in the soil profile of the overwintering population. Here in the x-axis, we have the abundance of cocoons. In the y-axis here, as represented by this illustration, we have the layers of the soil profile in it. So as we could see here, we do have a kind of similar pattern along of the years. In 2019, thanks to Tainari, to Deborah Montezano for carrying this study and collect those samples, process this data. In 2021, we got a kind of weird year. So we got a few cocoons from those samples. In 2022, we got a little more, not as much as 2019, but we still can go ahead, we're still getting the same pattern along of the years. We're having the first 3 layers in this first 2.3 inch in the soil profile, most of the cocoons concentrated, the soybean gall-mead concentrated. So jumping here into the temporal, the abundance along of the growing season, we performed another study where we had in a couple of sites plots of 20 by 20 feet, and then we collect along the growing season a couple of soil samples from those plots. We can go ahead. And here is a kind of illustration from what we had in the field. So we had the plots of 20 by 20, and weekly we collect 7 cores of soil samples from each of those plots. We process them and count the number of cocoons per week. You can go ahead. So here is an illustration of the results that we got from 2021. So we have 2 fields here. Each of those fields are being illustrated by one of those colors, all orange or blue. You can go ahead. So here we have in the x-axis the length of this collection from June to October. In the y-axis, we have the abundance of cocoons. As we could see here, you can go again, Justin. Yeah, we have kind of, we could identify some peaks of cocoons along of gall-mead along of the growing season. In this blue field here in Memphis, we were able to identify 2 distinct peaks in August and September, at the middle to end of September. And in Davie, we were able to identify one main peak, but we didn't get the same peak at the end of the growing season. So let's see what we got in 2022. So here in 2022, what we basically did the same thing, but we had 2 more set of plots at the end. So instead of having only 3 plots, we had the same length of plots, but now 9 plots instead to just 3 along of the field edge, covering each rows, each of those plots. Here is a kind of illustration of those plots, and we collect weekly 7 samples from each of those plots. We process and count the number of cocoons and larvae found in those samples. Here will be the same kind of graphic. So we have in the x-axis the length of this collection, and the y-axis we have the average of cocoons. And here, as we can see, we had a pretty different pattern from last year. And instead to get those distinct 2 peaks, one at the end and one in the beginning, the other one at the end of the growing season, we kind of got here a couple of weeks. You can go again, Justin, thanks. We kind of got here a couple of weeks with a pretty high abundance of this population instead to get those 2 distinct peaks. And here at the end of the growing season, we got this decline of this population. And here to conclude, I'm kind of not being able to see the end of the phrase, but yeah, to conclude here, thinking about the depth study, we got the kind of same result along of the years, where we got the greatest population in the first 3 layers, so 2.4 inch in the soil profile. Now thinking about the seasonal abundance, we got that this population is not even throughout the growing season. So this population changed throughout the growing season. And in 2021, we were able to identify peaks of this population, distinct peaks. And in 2022, got some pretty different results. We got a delay in this detection of soybean gall midge in the soil. And we got a kind of wave pattern instead to get those distinct peaks. What can be a result of this change that we did in the design? So we collect a little more further in the field. And the other thing is that at the end of the growing season, after the harvest, we got this decline in this population in the soil. With that, I think, thank you very much, everyone who are involved. And if you have any questions.
Okay, I do have a couple questions. One is for you, Mika. And it's with respect to those pupae in the soil, larvae in the soil. What is your thoughts? I know you haven't a study looking at this yet, but what are your thoughts on mortality of those larvae or pupae in the soil, both during the season and then maybe overwintering? Do we get a lot? Do you think there's a lot of mortality or differential mortality depending on when? What are your thoughts, sir?
That's a pretty interesting question. Thank you very much. And to be honest, I'm not sure how to answer this properly because we collect those cocoons. We have collect them, like to collect them, they go through some solution. So we have been trying to emerge them and see if there would be viable cocoons to put them in colony or try to do some study with them later. And we haven't get basically any emergence from those cocoons. We try to relate a little bit this population with the emergence that we have. We have been watching the case, but it has been pretty confusing. In years that we get like pretty high emergence of adults, we got lower number of cocoons being collected. And this year, for example, we collect a bunch of cocoons from the samples. We had a pretty weird emergence of adults from last year. So I'm not sure if those cocoons are just remain there, if they're dying, if there's something eating them. So yeah, about the mortality of those individuals in the soil, I'm not sure how to approach them this question and say if they're like going through this step like fine and become like emerging or if they're just dying.
Okay, I know you, there's another question. I know you haven't really looked specifically at soil moisture in your study, but what do you think there are any potential effects on the abundance or survival of the larvae or pupae in the soil depending on moisture level, dryness, or wetness? And that also relates to other questions of, does a dry year or a dry spring or a moist spring have an effect on emergence and infestation? So what are your thoughts there? And Justin, you can kick in too, of course.
Thanks for the question. This is a pretty good question. And when we see any other study about the population of midges in the soil, of gall midges in the soil, we could see in a bunch of other studies that the moisture have a pretty intense reflection in how this population will be susceptible or not in the next step in the emergence. When we have soils with a high saturation of water, this impacts a lot negatively this population. About the soil type, when we had here some soils that are more sandy or not so having clay, was easy to collect them, and we found a little more a high abundance when compared with like a rocky or like just a clay soil. My thoughts on that, like based on the studies that I've been reading, I'm pretty sure that the moisture can affect pretty badly the excess of moisture. And on the other hand, when it's too dry, can affect as well. And Justin, please, you can go ahead and help me with this.
Sure. Yeah, we don't have a lot of specific studies on that type of work. So I think Mick is going from the observations that we've seen, which is quite reasonable where we are with soybean gall midge. We did rainfall, artificial rainfall studies with a student in the lab last year and saw increased emergence. So that affects emergence. We used to see post rainfall events an abundance of larvae on soil surfaces, but that some of those observations haven't been consistent year to year. And so I think we also see larger cracks in the soil, which may affect where they are in the soil. So there's a lot of unknowns, I think, in that category. Good, you did good,
Okay.
Mick.
Here's another question that, you know, Justin or Aaron or Bruce Potter, several of us could comment a little on because we've looked at a little bit, and that has to do with tillage. Have you seen this have any effect on survivability or emergence or anything like that between no-till versus tillage?
I think Aaron has a talk coming up on that too. So
Yep. So we can wait for Aaron's talk, I guess, but that is on people's minds, so. And here's another question. Sorghum midge, someone mentioned that sorghum midge can set dormant in the soil for up to 2 to 3 seasons. And what do we think about the soybean gall midge? I would say, well, you know, maybe, don't know.
We've done a couple years of tracking soybean fields from 2 years ago. Micah, you've done that with soil samples. We've done that with emergence cages. We do not detect them in the soil 2 years out from a soybean field, and we do not detect emergence 2 years out from soybeans either. They could still be out there in really low numbers, but they are in very low numbers.
No indication yet.
Yeah, no indication yet.
And this is a different genus than the
Oh, yeah.
sorghum midge or the wheat midge in North Dakota. So it might have a
Yeah.
little different biology.
Yep. Okay, those are the
Yeah, we're not doing a very good job of keeping on schedule. So let's move on.
okay.
Our next speaker is from University of Minnesota, Phelan Anderson. Also going to be talking about the overwintering stages of soybean gall midge.
Hi, everyone. My name is Phelan Anderson. I'm a grad student here at Bob Cook Soybean Entomology Lab, University of Minnesota. And today I'm going to present some of my first year of research into the cold tolerance of soybean gall midge. So to start us off, why are we looking at cold tolerance? Well, most insects are cold-blooded, so they can't control the temperature of their own body and have to rely on the temperature of the environment surrounding them. This can end up being a pretty major factor determining their range, meaning it's important to understand how they can go through the winter and how cold they can ultimately survive in order to predict range, severity of outbreaks, population dynamics, things like that. For soybean gall midge, we know that they overwinter as larvae in silken cocoons in the soil, but we don't know anything about what kinds of temperatures they might be able to survive. In order to try to answer that, September 6th last year, we went down to Luverne, Minnesota, here in southwest Minnesota, and collected a bunch of infested soybean, brought them back to the lab. When they're in the lab, we basically needed to trick them into thinking they were going into winter. So the first thing we did was, in order to get them to build those cocoons, we dissected SGM larvae out from the stems and then put them in vials of sand so that they would start cocooning. After they cocooned, they still needed to think that winter was ongoing. So we split them out into 2 temperatures, 55 and 37 degrees Fahrenheit, and set them up with a shortened day length of 10 hours. And then after they were in those conditions for 2 months, we pulled them out and tested their cold tolerance with 2 experiments. The first of which was to determine their freezing point. To do this, we attached the cocoons to wires that record their temperature over time, and then cooled them down until we saw evidence of freezing. In this case, that evidence came in the form of these releases of heat that happen when ice crystals start to form. So if you take a look at the graph on the left, you can see temperatures going down over time until we see a spike, and that tells us that freezing just happened at that temperature. For our results, I'm going to show on the y-axis the mean freezing point that we observed for a given storage temperature that they have been kept at for the previous 2 months on the x-axis. And we can see that the mean freezing points range from around -8 to -12 Fahrenheit. We saw that there was not a statistically significant difference between our 2 storage temperatures as well. Next up, we want to look at the lethal temperature. So in order to do this, we want to actually find out what temperature they die at. We cooled them down to one of these 5 temperatures, similar setup. And then once they reached that temperature, we pulled them out, brought them back up to room temperature, waited an hour, and then dissected them to see if they're alive or not. So on the left here, you can see one of those cocoons that's been dissected open with, in this case, a dead larva inside. And for our results here, on the y-axis, we have the proportion that were found to be dead after an exposure to one of these temperatures on the x-axis. This is split out by color depending on our storage treatment. And then you're going to see dots that represent the actual proportions we observed, and then lines which come from our fitted model with confidence intervals. So what we see here is a pretty drastic increase in mortality between -4 and -13 degrees Fahrenheit, going up to 100% mortality by -22. Interestingly, if I add in a line which represents our overall mean freezing point, it aligns pretty well with that region of high mortality at around -10 Fahrenheit. Once again, we also did not see a statistical difference between our 2 storage temperatures prior to the experiment. So this is giving us some information on what temperatures they freeze at and what temperatures they die at. But what does this mean in terms of temperatures they might actually experience in the field? Well, we were able to get a hold of some nice soil temperature data from collaborators. This is coming from Grand Forks, Morris, and Lamberton, so kind of a north-south gradient here. On the y-axis, you're going to see the coldest temperature that was observed at a 2-inch soil depth for a given month. And that 2-inch depth is kind of the bottom end of where a lot of them would be overwintering. And the different dots represent different years' observations of that coldest temperature for that month. And these were all the most recent years we're dealing with, were up to 2022. Now I'm going to add a line that averages all these years together. And finally, add in a dotted line here that represents the average freezing point. And we can see right away the soil temperatures just never really get that cold. So our initial research here suggests maybe SGM is well suited to Midwestern winters, but what do we want to do next? We want to repeat these same experiments this upcoming year, just get another year of data. We want to add a lethal time experiment. So we know that the soil never really gets that cold, but they are staying at those temperatures in the soil for months at a time. So we want to pick some of the temperatures we used for our lethal temperature experiment and just hold them there for longer and see what happens. And then finally, we want to look more closely at the relationship between freezing and death. We have some initial evidence that freezing equals death based on the overlap in our freezing points and our high mortality temperatures, but some more targeted experiments would be nice to get at that more directly. And that's what I got. Thanks, everyone, and ready for questions.
We don't have a direct question about the temperature. We do have a question about where the cocoons have been found in the soil, about how deep. It's quite shallow, actually, usually less than an inch. Mika or Justin, if you want to be a little more precise on that, where you found them.
I think that question was like, how far down have we found them?
Yeah, how deep in the soil have they been found, and where would you say most of them are? That's my part of the question.
Yeah,
Well, that was in Mika's talk.
Yeah,
showed
but that's
that
the only
data.
question we have.
Okay.
I think it was difficult to see the depth, but they're down to 5 inches. We just don't know if they're viable down at those depths or just detected.
Yeah. Okay. Well, let's move on to Aaron Hodson, who's going to be talking about preseason tactics.
Thanks everyone for joining. I think if the schedule is correct, we have a short break after this just to kind of maybe catch us up and stretch legs if needed. But there's already been a couple questions about maybe how tillage can influence soybean gallmage. And so I just wanted to give a little snippet of some of the research that we've been doing. And I say we broadly because it's mainly been coordinated by a graduate student at Iowa State University, Ben Colby. And so I just wanted to acknowledge him. I think he's maybe actually on somewhere, but also with the help of Justin, we've tried to get a better understanding of some preseason management tactics. And so let's see here. When I think about all the possible tactics that farmers could use to mitigate or suppress field crop pests, this is a really common graphic of all the different components that could be possible. I think we're going to hear a little bit about biological control and genetic control when it comes to host plant resistance today. Usually the big hammer in field crops, the go-to thing is chemical control, but also a really important component of some systems is a cultural or mechanical control. So when I think of cultural control, it's really things that are in the hands of farmers, things that farmers are doing to have the healthiest, most vigorous growing crop that they can and get, the high yields that they want to at the end of the season. So tactics that farmers can use to maybe discourage pests from being so successful would be things like data planting, just a final plant population that you're hoping for. Even things like row spacing and weed control are really important to either help or hurt insects. And so that's something we think about quite a bit. And then things like crop rotation. So making the plant less available, especially if the insect isn't mobile. And also tillage, I think, would fall into the cultural or mechanical control, things that are in the hands of farmers. So just trying to learn a little bit more about how tillage can impact pests. Basically, tillage disrupts the soil. And through the literature review that Ben and others are doing, it overall, tillage or the disruption of soil tends to benefit pests. And that sometimes just the mixing of crop residue with soil, it can influence soil temperatures. So especially the top 2 inches of soil would warm up faster with tillage. And soil moisture could maybe help or hurt insects. But basically what tillage is doing as it's disrupting the soil, it's either bringing insects closer to the soil surface or moving them down deeper. This could be an advantage or disadvantage. And in the case of some tillage practices, you may actually be physically crushing or killing or injuring the pests. In some cases, the disruption can cause desiccation in certain times of the year, or even just bringing them closer to the soil surface, you could get some UV exposure to those pests. But a lot of times I think about tillage as reducing the weed pressure. So for some insects that really rely on weeds for a refuge or a pollen source, taking away the weeds is a real disadvantage for them. And then in some cases, it can also alter the natural enemy complex. We'll hear a little bit more about that later, but it could also help or hurt some of the predators, pathogens, and parasitoids that we see in field crops. So more specifically about this project that Ben has been doing for 2 years, I specifically want to talk about what he did last year. He worked on a commercial farm near Wall Lake, Iowa, and he had 4 different tillage timings. He had a fall treatment, spring treatment, fall and spring treatment, and then he had a no-till treatment. And if you're super into tillage equipment, as far as the type of tillage and the depth that he was able to achieve, you can see here he had a disc and a chisel, and he had different depths he was able to achieve in the fall and the spring. So just a couple pictures that he took just as he was tilling the very edge of this field near Wall Lake. To evaluate the responses to these different treatment types, we use these Illinois-style rootworm cages, and he put a couple in each plot, which were replicated. And there's a little canning jar at the top, if you're familiar with the rootworm world at all. And so as the adults come out of the soil, trying to find new locations to mate and lay eggs, they basically are collected into these canning jars. And so he would look at these treatments at least twice a week, and he was monitoring the overwintering emergence. So right as corn wasn't planted yet, but eventually these cages would be mixed in with corn as they were trying to emerge and then move to soybean. So the super sad and super short answer to this question from the 2022 project is that he captured very few adults. And I can't tell you how many people hours this was. But unfortunately, he only captured about 19 adults through all the treatments over a very short window of emergence of 7 days. So you see on the right, there's some differences there, but if you notice the scale of the adult captures, it's not even a quarter of a midge. And so the response rate or the capture rate was very, very low. And so in my mind, it's really too early to tell if tillage has an impact, whether it's helping or hurting soybean gall midge. I think it is influencing the overwintering success in some way. I just need to establish a better way to collect adults and maybe have a field that has a little bit more pressure so that we're actually able to compare the treatment implements and also the treatment timings. And so
To you, but I'll kick it off with some kind of an overview of adult emergence of soybean gall midge for the last couple of years, and then we'll take a brief break for questions and then go on to some insecticide work that's being done. So obviously a bunch of authors again on this slide because there's a lot of us tracking adult emergence, and as Aaron Hodgson's mentioned, it's pretty difficult to track adult midges in the field. So this is probably, of all the information on adult emergence, one of the most important in terms of the challenges we face with the upcoming discussion on insecticides that have residual activity, but not as long as what's indicated on this particular slide. So the first part of this is when they've first been captured for emergence. So we were mid-June in 2019 and 2020, and then we've been detecting them May 31st and June 2nd here in 2021 and 2022. Usually once they're detected at one location, they follow to others pretty quickly. And I'm not going to provide a ton of detail because we are limited on time, but all this work is funded by North Central Soybean Research Program, as well as by our state boards in some of the states for increased capture sites. This duration of overwintering emergence is a really important factor for soybean gall midge. We initially started right around 15 days on an average. That seems practical for a well-timed insecticide, and then went on this nasty trend upward, averaging about 35 days in 2021. If you go back to those conversations when we were looking at this really wasn't feasible for any insecticide to last that long. And then in 2022 here, a bit of reprieve. We're back down, around that 24 days on average, still some long and short sites in terms of duration of emergence. And all this duration is all from last year's soybean fields. So a couple important things. We can estimate the days per generation until we develop a colony and can do a lot of testing in the lab and learn more about this insect. We can make some estimations from the field. And so to make those estimations, if we determine the first date of capture from an overwintering site from last year's soybean fields, and the first capture from this year's soybean fields, that time between those two events provides us an understanding of adult activity to adult activity again, and the understanding that they have now went through a generation. So if we look at that time frame and the number of days equaling one generation, got surprisingly consistent trends that we've seen over the last couple of years of 26 to 29 days between each of the years in terms of the average between those two capture points. That to say that the range is actually between 18 and 45 days when we do capture adults in both sites or track both sites. So there's quite a bit of a range around that. This is 2022 data summarized across a large number of sites. You can see the very low numbers, as Erin mentioned in her talk. Soybean gall midge went from an average adult or total adult capture in 2020 and 2021 of about 5,000 to 6,000 adults across the network to right around 1,000 in 2022. And why that happened, we don't know. You've obviously seen the survey, so we still have presence of gall midge, but it's a tricky insect to follow. But regardless of that lower number, it's the same trend as in previous years. We have kind of this buildup of adult emergence around mid-June that's the same each year. It declines and tails off well into July, where we see low levels of emergence with the last detection usually around mid-July in fields. And then soybean gall midge begins emerging from this year's soybean fields. There is a tiny little bump right on the 1st of July there, about the 1st. And then they climbed up and we had kind of a bumps in populations throughout the remainder of the season. with in almost all 3 years, around mid-September is our last adult emergence for the season. Coinciding with this, Vilma Montenegro did a lot of work over the last couple of years looking at larval abundance. This is her last year of studies from 2021. And you can see that from first adult detection, we pick up larvae. The populations remain relatively low in that first group, and then around late July, early August peak, and then go into kind of a steady decline for the season. I mention this because this is a really good time to scout fields in late July, early August, at least for this Nebraska data. That's typically when they're most abundant. Okay, one of the main questions we get about adult emergence, adult activity is how far do they go? I'm just going to show you 2 slides on larval presence, abundance in the field from an overwintering site into this year's soybean field. So these are collections occurring from the edge of the field in the current year's soybeans. Our last year's soybeans is just over here, and it goes all the way out to 500 feet. Lots of data was collected on this. There's a presentation available in the previous ones that talks about injury and yield, but we're just going to focus on larval number for the sake of showing where they are in the field. So this first sample is distance from the edge of the field on the x-axis and larvae per plant on the y-axis. And you can see that larvae are most abundant right along the field edge and decline with the population declines as you go out in the field, but we could still find them even out at 500 feet into the field. And all of this presence of larvae that you see here is all related to overwintering generation adult movement from the adjacent field. If we come back a month later and sample again, and now they've gone through a cycle on the current year's soybeans, they've reemerged and infested those soybeans. Now you can see a much greater population along the edge, and this reflects the amount of injury we see at the edge of the field, and then a decline to kind of a consistent but lower number here, even out at 500 feet from the edge of the field. So gall midge is primarily present right along those field edges. So the take-home message is we saw a lot less adult activity in 2020. I think this relates to some of the new things that you'll be hearing towards the end of this webinar about the use of pheromones and identifying a pheromone, and Ian Kesey is going to speak to us about that. But that troubling trying to track this insect, the long duration of adult emergence, that's key in the management difficulties with insecticides, especially coming from last year's soybean field, likely 2 generations occurring. The generation time is somewhere between 26 and 30 days until we have a colony. And the greatest larval abundance is right along that field edge, but they can be found out to 500 feet from the field borders. So that's what I have on adult emergence. Any questions on that?
Well, actually, we had a question in the chat for Erin from her previous presentation on tillage.
Let's take that one then.
Erin, did you see the question in the Q&A?
I attempted to answer the first one, and I'm having a little technical snafu with the question that Marty, I'm trying to answer it, but I'm having a little bit of a snag. But I did, someone asked about the sort of the low replications, and I totally agree.
Yeah.
It's really hard to sample adults coming out of the ground with the technology that we have, which is old rootworm traps. So.
Okay. Well, we don't have any other questions right now. Justin, why don't you go ahead with the next talk on insecticides, I believe, right?
Yep, I put it in the same slide set.
Okay.
So
and I'm stepping on Tom Hunt's toes. He should be moderating, but
That's okay.
yes, you can cover the next ones.
All right. Well, I'll cover insecticides. I think, as Erin mentioned, this is what a lot of people lean on when they run into soybean gall midge, and so I'll provide a pretty broad overview to this topic, show a little bit of data, but mostly focus on, I think, a lot of the challenges we're facing with insecticide use. So I've divided this slide, this talk up into just a couple slides to show you everything that we've applied against soybean gall midge that is commercially available, kind of some highlights that illustrate some of the points that we've seen. This first slide just shows 3 different classes of chemistries, group 1s, 4As, and 28s, and some of the chemistries within that have been applied. If you're interested in specifically what was applied, we could certainly go through that, or you can ask questions. But those are the different chemistries that have been applied. Of those, we've just not seen a ton of consistency. It's been difficult to get consistent results. Probably one of the few exceptions, and it even has its rules of inconsistency, is Thiamet. This is from the first study that we took on this, which was at Davie, Nebraska area. And you can see we got reductions in larval number, and then a pretty clear trend between increasing rate and yield for a high pressure field with soybean gall midge. That's been seen in subsequent studies. Other studies on neonicotinoids as seed treatments, thiamethoxam, imidacloprid, and clothianidin, some very common ones. You could see that, you know, they have some effect when you look at reductions in larval number and increasing rates of, say, gaucho, and similar things have been found with, you know, thiamethoxam and Cruiser Max. But then when we get down to yield, we tend to lose those differences in a lot of cases, or have difficulty detecting them simply because of likely because of the long duration of adult emergence and the difficulty with controlling something that, you know, doesn't show up really in our plants until V2 stage, when those fissures start to occur. There's a whole list of foliars for you to look at, you know, some organophosphates, as well as neonicotinoids, butanoids, pyrethroids, spinosins, and diamides, and you can see the range of chemistries under those that have been tested. You know, we've had some luck with pyrethroids, but again, this group falls in the same group as previous, which is we just don't get a consistent result across sites, or even within, you know, different sites in the same year. Here's an example. You know, I think you may all think that we're just out here to frustrate you with some of the data that comes out, but this is one example, the use of drop nozzles. These are drops that come down in between the canopy, which I understand, yes, is relatively small, but they allow the spray to go horizontal against the plants and potentially hit the base of the plants, and leverage, which is a neonic pyrethroid, showed increased efficacy or reduced larval number when we use those drop nozzles relative to an overhead application. So one example of the timing and placement, or the placement of those chemistries. And this just follows through. I know you'll see AUSPC a lot in this talk that Mitchell Helton did the work to show area under the severity progress curve. And its numbers. Basically, the higher the number, the greater the injury. And here's just another example where, you know, we apply a chemistry, we get reduced injury on those plants, both, you know, mid and late season. But then when we get to yield, we see sometimes a small numeric benefit, but, you know, you'll realize the yields are pretty low under this scenario, indicating there's a lot of pressure in that particular field. So I'm going to leave with this slide as the challenge we face with insecticides. We have this long duration of adult emergence, which should be burned into your memory at this point. We have season-long adult emergence, and if you hold off for a presentation or two, we'll show you, you know, the challenges with that in protecting plants. Adult behavior, they're not existing up in the plant canopy, at least by indications of sweep nets. They're down near that soil surface or around those points where they're infesting, and they don't live very long. And so they're difficult to get to if you're targeting adults. Larvae feed within these protected areas of the plant. You know, the eggs are laid just inside these fissures and feed underneath, you know, the outer layers of the plant. And all life stages are present in the field, you know, from Mika's work and the larval abundance work. You could see that, you know, we have cocoons present all, you know, from when we first find them, along with all stages of the larvae as well. So it makes for a very challenging environment. So with that, I'm sure there's a couple questions or floating around there on insecticides.
We have a few minutes, about 5 minutes, for any comments or questions.
There's no questions in the Q&A yet for you.
No?
Okay, here we go.
Something popped up.
Yes.
I can
Can you see that, or?
I can see it, yeah.
Okay. Well, I'll repeat it for everybody else. Did the use of rootworm soil insecticides in corn have an effect on soybean gall midge? Or you think whether less use of soil insecticides in corn maybe benefited soybean gall midge?
Man, good question. We don't have any direct data towards that. I'm not following growers that are using soil insecticides in those fields, so it would be difficult to know directly. And we're not obviously running tests on soil insecticides that are for use against rootworm. Others have comments on that question? That's a tough one.
Yeah, we don't have data. I guess got a different question came in. If a grower was to use a drone, what would be your recommendation on insecticides and timing?
We've used drones as with insecticide applications. A group of us has a drone. The timing was the same as ground applications. We actually saw some increased efficacy from down blade rotor pressure, but that was done in 2019. And I guess I would caution 1 year of data and the inconsistencies we're seeing anyway. But those timings would be relatively similar or the same as our timings that we've suggested, but haven't seen a lot of good responses with insecticides.
Okay, that's all the questions we have in the Q&A chat right now. Excuse me, one more, several more. Any work on biologicals that have efficacy against gall midge?
I think we'll have a biological control talk coming up. As far as biologicals for sprays, yeah, we've had conversations. I've never done a nematode application or anything like that. Others, if you want to chime in on that, there's been lots of conversation, but
And I guess one other question, not related to insecticides, but do crop rotations have an impact on soybean gall midge, and any recommended rotations?
Yeah. Soybeans next to soybeans each year obviously allows gall midge to move back and forth between fields relatively easily. Breaking up that rotation with other crops is a good way to reduce the midge. It's the landscape level of that process that's probably more complicated because of all the bordering fields. When we get into conversations with growers, that's the challenge. But we see rotations of fields within a farm so that they're not back and forth between corn and soybeans as a control measure that seems to be effective.
Okay, I think we've kind of run down on questions. And any that remain can be answered later in some of the other sessions. So I think we're going to move now to our next speaker. We'll be talking about hilling and unhilling, the effect that has on the soybean gall midge. Pragya Gupta will be leading that talk. She'll be joined by Justin.
Hello, everyone. Thank you for joining today. And I'm a master's student at Dr. Justin McMicken's lab, and I will be talking about the management practices for the soybean gall midge, and more specifically, I will be focusing on the cultural approach of hilling. So before talking about hilling, from the previous talks, we have pretty much known that the fissures that starts to appear at the V2 stage are the cause of the entrance for infestation. And because the soybean gall midge has more than one overlapping generation, the gall midge has the potential to cause a season-long infestation. And the focus of this study to understand the crucial stages or the timings when the soybean needs to be protected from the gall midge infestation. So this was performed by doing the hilling study. And what is basically hilling? So hilling is placing soil around the base of the stem, and for this study the soil was placed at the base of the stem where fissures occur up to the unifoliate node to create a physical barrier to the gall midge, restricting its ability to reach the fissures and cause infestations. So the objective was to determine the impact of timing of hilling on gall midge infestation, plant injury, and the yield. The study comprised 3 hilling timings based on the plant stage and an untreated or the unhilled as the control. You can see these are the different stages when hilling was applied, and these were the 3 different timings when hilling was done, and there was an untreated control. And a total of 4 samples were collected during the season for different evaluations. The methodology for the larval abundance was the plants were collected from the row 1 and 4 and they were peeled to check for the larval abundance, and up to 3 infested stems were collected and brought back to the lab for counting the orange and the white larvae. So the results for the larval abundance showed that the treatments that were held, those are the treatments that were held at the V2 stage, the V5 stage, and the reproductive 2 stage. So those treatments had the lower larval abundance compared to the treatment that was not held. And also the V2 stage that was held earlier in the season had the lowest larval abundance compared to all the treatments. The data was also collected for the injury scores and the yield, and for the plant injury scores, it was done based on a visual assessment, and the yield was done at the end of the season from the middle 2 rows. The results for the injury score showed that the treatments that were held had a lower injury compared to the treatment that was not held, and it was significantly lower injury in the treatments that were held earlier, that is the V2 treatment. The results for the yield showed that the treatments that were held had a higher yield and more bushels per acre compared to the untreated treatment, and also the R2, it was still lower than the untreated. So from the results for the injury scores and the yield, we can see that the hilling treatments performed better than the treatments that were not held during the season. Overall, the summary of this study was the larval abundance was impacted by the timing of hilling. Hilling at the V2 had the lowest larvae number per plant, and the V5 and the R2 were lower than the untreated. The greatest plant injury occurred with the no hilling treatment, and only the V2 timing had the lowest injury compared to the untreated. Talking about the results for yield, the yields were significantly greater for the V2 and the V5 treatments compared to the no hilling. So for all the results, we summarized that the treatments that were covered or held during the season performed better than the treatments that were not held, and also the treatments that were held earlier in the season, that is the V2 treatment, performed better compared to when hilling was performed at V5 and R2. The significance of this study is we know that the soybean gall midge has a long emergence period, and it emerges from early June until mid-September. So although hilling isn't a practical strategy, it is important in understanding the management. The stem fissures are critical factor for the adult emergence and plant injury. So from the hilling timings, we observe that the control tactics need to be applied earlier to be effective. However, highly effective strategies may still have value when they are applied in the vegetative stage, as we can see, even if the hilling was done at the V5 stage, it performed still better than the R2. So to have better results is always preferred to perform the management strategies earlier in the season. The no significant differences in yield was observed at the R2 stage. So we can see that hilling is a potential strategy for growers that are under significant pressure. However, the study will again be conducted this year, 2023, to have more results and more observations. And thank you. And if you have any questions, I will be glad to have them.
Okay, we had a couple of questions for Pragya. One is, how common is it for growers to have implements that would hill soil to the unifoliate stage? And what row spacings would this be an effective practice for?
Sorry, I think I was mute, so I didn't.
Did you not hear me?
No.
Now you can hear.
Yeah, now I can hear.
Okay. The question was, how common do people have types of equipment that could hill soil to the unifoliate stage? or unifoliate leaf, and what row spacings would this be most effective to use? And maybe Justin can jump in too.
Uh,
If you want me to answer it, I can.
Sure.
Yeah, it's a tough one, Prague. Yeah, so because I ran the implement on your study, they're done on 30-inch row spacings. Doing it on narrow row spacings would be pretty tough. Hilling to a unifoliate, area on the plant when the plants are a V2 is very difficult. And certainly if there's any change in terrain, it's hard. So it's not a practical thing in terms of management unless you're a grower that's under a lot of pressure, and this is one of the few strategies you have left, and it's viable for you to do. We explored delayed timings of applications because those become more practical and easier to do.
And then the other question is, what is the impact of strip tillage or ridge tillage on gall midge emergence?
Bruce, I think we should defer to Bruce on that. Bruce, you see a lot of ridge tillage up in your area or others. Not ridge tillage, sorry, strip. Strip
not
tillage.
so much ridge till anymore, Justin. I can tell you the farm that we see the most consistent gall midge problems with in Minnesota, that I know of anyhow, is strip till, but I'm not willing to say that it's because of the tillage system. So I can't answer that.
Yeah, sorry, Bruce. Ridge tillage is down in our area. We have a few areas that have a lot of ridge tillage. That's how we came up with hilling as a potential strategy. We don't see them in ridge tillage, but it's done much later in the season to where the fissures are covered.
Right.
They plant on a ridge early in the season, but it doesn't.
The difference with the strip till is you've always got an area that's undisturbed soil, so you're not disturbing larvae or pupia, at least in between rows, but otherwise, I don't know.
Another question is, does having a thick mat of residue such as irrigated corn residue or cover crop residue give any benefit similar to hilling in terms of covering up the base of the plant?
Yeah, sorry, Froggy, you're getting all the tough questions of things that are.
For this, I think having that crop residues, like people think that can also act as a hilling, but in my view, that may also be another area for the pest to overwinter. So that can increase the pest, like the other pest can hide between them. So it is not equal to hilling, because hilling is creating a physical barrier, and those crop residues can provide other disadvantages in addition to that thing.
Okay, here's a question maybe Praga can answer. How many sites was the hilling done at? It was just one site?
One site, yeah.
Okay.
We've done, we did it at other sites the previous year.
Yeah. And another simple question, how does planting depth have anything to do with fissures?
That's more complicated than I think
Yeah.
we all think. And I know George Graff and I have talked about this, so I might let George answer that, or have George give his best thoughts on that later.
Okay.
Okay. I think we have finished most of the questions now. Any others we can move to later. I think we can move on now to the unhilling section that you and that Justin and Froggy are going to present.
Okay, I know this hilling is pretty frustrating for people to listen to, but think of it as a tool of understanding, kind of in the same way that Pragya presented the last one. So this is also Pragya's study. I'm just helping her out from doing back-to-back talks. But this is a bit of a different look. And so I'll creep through the introduction piece of this to show you how we set this up. And so a bit of background on soybean is most, if not all of you are aware, there are yield components that are formed throughout the season. Nodes begin to appear around, as soon as the plant really comes to the ground, we begin to accumulate around the VC stage. And then they'll continue to produce new nodes through the R5 stage, which is this beginning seed development. We can get branches, and flowers are so abundant, they're less important, but we need them, obviously. Pods begin right around late flowering, just on a different part of the plant to continue through R6. Our seed size or number is determined between R4 and R6, and then seed size is right here at the very end. So why care about growth and development and yield component setting in soybeans with soybean gallmage? Pragya and I designed a study to look at fissure exposure of soybeans during the season to say if gallmage had access to plants the whole season and then limited amounts, what impact would that have? So we held an entire site at the V2 stage, so just as the fissures started to appear. And then in the case of this first treatment, we immediately uncovered the plants and left them exposed. And this red line that you'll see as I add the other treatments, it was exposed through the end of the season. We then did another one that of course was covered at V2, but then uncovered at the R1 stage. And so this gives a protection period through R1. You could look at this as if we come up with really good tactics and could bridge this gap with more complete control, that would be the control. And then we have the fissures being exposed for the remainder of the season, and so on and so forth for the remainder of the R3, R4, R5, and R6. And Pragya painstakingly uncovered all these plants by hand. And then we have one treatment left where we never uncovered and left them covered throughout the season. And so the question exists, at what point of the season do we not have impacts from soybean gallmage? So if we can protect plants to these different points in the season, at what point do we not measure a yield impact from this insect? So I'm going to go right to the results. There's a lot of work that went into these results, but when we uncovered plants and Pragya went out 2 weeks later, she quickly picked up infestation in those plants. So within 2 weeks of uncovering and exposing fissures, we rapidly got an infestation of soybean gallmage. And what you see here is the average number of larvae per 3 plants on all those trees. So V2, 2 trifoliates present, full flower, the first, stages or first part of early pod development and late pod development all have pretty significant numbers of larvae present on them. And then our later pod and beginning mature, late pod and our beginning seed and late seed have relatively low numbers that are pretty close to a completely covered plant. So obviously uncovering earlier impacts that process. AUSPC, again, Helton's study from 2022, injury, the higher the number, the greater the injury, a pretty nice relationship between those that are exposed early and for most of the season to a decline in late season, very similar to larval number. They have a very similar response. If we go to the actual yield components where we saw impacts, you can see that the impacts are so heavy early in the season, we actually impact the node numbers. So we have less node numbers here, and then we fit into a pretty normal group here that's around 16 nodes per plant, even with the covered. As you move further into the yield components into pods, there's very few pods down to 10 to 15 pods or 12 pods or so on these earlier ones. And now we're starting to impact that R3 stage treatment where we're actually at beginning pod is a little bit less, but then things are pretty normal for R4, 5, and 6. If we go to seeds per plant, so the number of seeds per plant, as expected, less pods, less seeds. You could see this depression in the graph continually pushing down as we move into later yield components. But again, it seems to be confined primarily to these first 3 treatments. And the same thing can be said for seed weight in grams per plant, where we have relatively little seed available from these plants. I should mention this light color here that's on top. These are abnormal seeds that probably would get blown out of the back of a combine. So if you wanted to ignore those, you could. The dark ones are normal seeds within that set. I know grams per plant is a rather odd thing to think about. And so I looked at the plant stand and we calculated an average or approximate yield from those plants. So we're looking at 10 to 15 bushels up to about 42 or so, and then on up into the 60s for these remaining last categories. It's very approximate. And the differences amongst these treatments, this data is pretty new. So we have some work to do on the analysis. So Adults quickly infested soybean plants after fissures were exposed. This is actually the image from the field. This is one of those V2 treatments right next to others that remain covered. So you can just see how quickly they infested and killed off those plants. Large infestations of larvae occurred up to the R4 or late pod stage. Larval populations began to decline in mid-August. We also saw a decline in adult activity. Plant injury was steadily declined with delayed exposure, and the yield components were impacted, what appears to be through the R3 or early pod stage, which suggests a really long control window for this insect. So hopefully that gives you some insight into the length and time that we might need to control this insect to avoid significant impact. I will say one final thing is that this is a very high pressure site with corn on either sides last year's soybeans. So it represents kind of our worst case scenario for soybean gall midge.
Well, Justin, before we get into some of the other questions, I have a throwback question that came up with the insecticide application issue question that was asked earlier. At what time, if we were going to try to manage this pest with insecticides, what is the timing of that application?
Yeah, good question. I guess we covered these things in previous and then forget to cover them with each new session. That alert network, when we put that out, that's when we usually, people that are going to manage soybean gall midge typically do that. So at first adult emergence, and that's where we've seen some control. We don't want everybody spraying, obviously. We're coming into a conversation on biological control and what might be out there. So you've really got to have a presence and history of soybean gall midge in the adjacent field to warrant applications like that. And certainly not whole field if you're doing some sort of application. So I think that kind of answers the question, Tom.
Yep, thank you.
And relative to fissures, a question earlier, is it possible to develop a coating that can be sprayed to seal the fissure?
Yeah, there have been some studies tried on that. I think the challenge we face with spraying, and a couple labs have tried this, or groups from other states, is that stem expansion is constantly happening. So the cracking of that material has to be fairly elastic, or it may continue to crack and create open wounds, or open spaces for the adults to get in.
Yeah, I think we were thinking about the kaolin wax, something like that. But again, as Dr. Justin McMicken mentioned, that those cracks and fissures develop throughout the season.
Okay, it looks like we're done with the current questions. So we can move on to our next speaker, George Graff, the UNL soybean breeder, and he'll be talking about host plant resistance. Okay, we can see it.
Okay.
There we go.
All right. So, we're going to just have a quick review of where we were last year when we introduced genotype as a treatment, because you heard all about different kinds of approaches through management or other kinds of treatments to try to control this pest. So, it's not advancing for some reason.
If you click on the slide once, sometimes that helps.
There we go.
So where we were, this involves the great entomology group, and then Diego Jarquin, Luis Posadas, and I were involved on the host side for working with trying to understand response by the soybean. So just to quickly review, we looked at, we wanted to include as many genotypes as we could. So what we did with a study that we had in another North Central funded project was we looked at the USDA soybean germplasm collection, and here with that that rogue red thing roaming around, the gray dots are all accessions in maturity groups 1 through 4, just everything that's in the collection based on their 50,000 SNP genotype information. And you can see this kind of represents the spread and their diversity, you know, how different these are from these. And then we use 3 different sampling methods, a k-means cluster method, a supersaturated design, and a random, just random sampling. And with those 3 together, you can see we represented all the genetic diversity in the USDA soybean germplasm collection. So we went based on the data that Justin and the other entomologists collected in 2020 and 2021, we were able to do a genome-wide association mapping and find significant SNPs or genes or chromosome areas that were significantly associated with injury score on all or on 17 of the 20 chromosomes of soybean. So what does that tell you? First of all, that there's at least 17 genes and probably more involved in the plant response to infestation. So it gets complicated. But we also included in those years a small, well, actually 160 or so elite lines from our experimental lines from our breeding program and things that had been commercialized. And so what we saw from there is that the injury scores were skewed to the low end. You can see that even the highest injury wasn't that great, and that within that range of injury scores, there was a skew toward the low end of injury. So that was some good news. So we said, yeah, there's differential response to infestation. There are genotype differences. We mapped genomic regions and we identified significant SNPs for follow-up work. And it seems like, the good news, at least for farmers and companies, is that with that kind of information that the entomologists in the screening saw and uncovered in the elite germplasm, there seems to be potential for some quick response by companies if they can evaluate the germplasm well and both phenotypically and genotypically identify the best lines that might be suited for areas that are infested with soybean gall midge. So we wanted to follow up on that, and we tried to use that, that QTL information, those significant SNPs, like I showed on, 17 of the chromosomes, and try to identify those some of the lines from that 750 or so that were evaluated. We wanted to get a smaller group and do more work on those. But what we found, and this was more work by Diego, looking at 10, 20, 30 numbers of significant SNPs and relationship with the index or the injury score. And so there's really no correlation with those smaller numbers, 10, 20, 30, 40, 50, until we get around 1,000 or even 2,000 markers. We start to see some kind of significant correlation with injury score. And so, we did find we needed 1,000 or 2,000 or more to find a stronger relationship with damage score. And so that emphasizes the probable multi-genic complex nature of the host response to soybean gall midge infestation, at least in this group, the way we're evaluating it across that large number of genotypes. So in 2022, we used that 2020 and 2021 data like that I just showed you, and on those 750 PIs and went back to the soybean germplasm collection. And again, it still is kind of unique that the soybean collection has genotype information on pretty much every accession in the collection. So we use that in the first place to identify a group out of the 20,000 that are there. We got 750 that represented that diversity. Then we went back and projected, that injury score, the genotype information in the injury scores that we got from those 2 years of evaluation to predict the injury scores on all the untested accessions in maturity groups 1 through 4. And so just by using the genotype information and that relationship between the genotype and phenotype that was established in those 2 years, we could basically get an injury score for all the other accessions in the collection. So then we selected 100 of the accessions with the lowest predicted damage scores and grew those and increased during 2022 to use for this year's evaluations. And also in 2022, we had yield test plots in Iowa, South Dakota, and Nebraska, with a group of those accessions that were selected based on, again, that 2 years of data. And we included both accessions that were supposed to be, classified, or that were classified as resistant based on their injury scores over those 2 years, and included a few accessions that were more susceptible, that had higher injury scores based on those 2 years. And Justin took larval counts on the yield test plot borders at the Nebraska locations and damage scores on yield test plots that included multiple ratings at the Nebraska locations on 5 dates. And so we're going to, when we take a look at that, you see that, well, first of all, damage severity scores were made in July 14th, 26th, and August, 3 dates in August from the 5th to the 24th. And this is at Syracuse, which was a pretty, had pretty high infestation. And you can see here that there are 3 lines that have 0 damage, basically. And then there's about 9 lines that have really minimal damage. And this is just by chance. These, Nebraska experimental lines were just included as checks in the test. They weren't, the evaluation was these were included as yield checks, but even so, their damage scores are also lower than the susceptible commercial check. So the important things from here, there's a range again in damage and accumulated damage over the season, and there is validation that that these ones that we identified as susceptible, as resistant, are coming out again as resistant in the subsequent evaluations. And we saw similar results from the data that Justin got at Walton, Nebraska. It wasn't quite as a high infestation, but again, here's a susceptible commercial check. We have things that are worse than that. We have things that are better than that. And there again, there's a bunch of, or a few accessions that remain near 0. And there is a decent level of, lower level of damage in at least a couple of elite lines that were included in there. And then looking at larval counts, there was this is at 2 Nebraska locations in Syracuse again and Walton, and they're pretty similar. And then looking at results combined across locations for 2022, the points are there's a significant difference in larval counts. Here's the susceptible commercial check. There's and I think, the important point is that using the larval counts along with the area under the severity progress curve, we can identify, start to identify lines, a smaller group than the 750 for sure, to do more targeted and more detailed studies on, because again, we see significant differences in response, and these 3 PIs represent 3 different maturity groups, group 3, 4, and 2, and there are others in there. The thing that we noticed was, and I did check this, that lowest scores in the 2022 evaluations on those graphs were all identified as resistant based on the 2 years prior results. They weren't, none of these are susceptible, were classified as susceptible in those other 2 years. So we have some validation that, the ratings are identifying lines, and they're identifying lines well. And those, at least at the bottom end, those have been pretty consistent, or very consistent over years and location. Variation also exists among elite cultivars. So we do need broader evaluation of current elite lines from both public and private programs. That'll help get good information on elite lines in commercial programs and help with recommendations and for growers, and especially in areas that are most severely affected. And like I said, we can narrow down the number of accessions evaluated and get more detailed data on larval counts and plant damage and kind of interactions and what components, what things are most important in that life cycle of the host and the pest, and things that we might target, might be the best targets for identifying the best and more lasting resistance. So for this year, we're planning on evaluating that set of 100 PI accessions that we grew the increases of this in 22. Those were all, this set is supposed to be all resistant. So, then we can start to get into some, maybe on the smaller group as well, overlaying some of those, maybe some of the promising management options and start to look at interactions if you're more resistant and add this management that had some positive effect, could we have basically no damage. We're going to retest accessions for yield and reaction to gall midge, and then hopefully broaden the evaluation to elite high yielding lines. So thank you very much, and I can take a couple of questions if there's time.
We don't have any direct questions as of yet for host plant resistance, so I guess we'll move on.
Well, there was one question I passed on earlier that maybe Justin could answer. Is there any variation among soybean varieties in the number of fissures they have?
That's almost a George question rather than a Justin question. Go ahead, George.
Well, I was figuring you were down on your hands and knees looking at the stems more than George was, so maybe you noticed. Yeah.
Gallman showed up and George took more of an interest in doing the same thing. So go ahead, George.
Yeah, well, now that we can look at smaller numbers, we will definitely get more detailed data on that. What Justin said was that everything he evaluated had fissures. So I don't think that's been a major component of anything so far identified as resistant, which I think is kind of a good thing too, because it means that we have something going on in the plant that is maybe some antibiotic kind of resistance. And so those are questions to be answered, and we will definitely look at those 100 resistant PIs and see if there's any variation in their fissures. It might be a component of that, the things that were selected, because like I said, there's all kinds of genes on pretty much every chromosome that are involved in this response, and so one of them might be smaller fissures or no fissures at all. So, but we don't have the details right now.
Okay. We're running a little behind, so I think we can save the other questions till the end here. So our next speaker is Ian Kesey. He's a professor in the School of Biological Sciences, and he's been looking at pheromones with respect to soybean gall midge. So that'll be our next speaker, and so let's bring that up.
Excellent. Okay. So thank you first of all for the invitation. I'm in the School of Biological Sciences. I'm a relatively new faculty. My background is really in chemical ecology, insect behavior, and neurobiology. I work with sensory systems. I'm very interested essentially in how insects identify and navigate towards their host plant, or in the case of today's talk, really how they are identifying each other. So in this case, really how males and females, for example, are finding each other, and that's primarily through pheromones. So I would say this is a relatively new project, or this is just a starting project. So I'll mostly be talking about the direction and goals for this project. There's some preliminary data, although it's very early. So I'll just say that there's definitely some promise that's showing here. So maybe I'll just start, depending on everyone's background, what is a pheromone? So in this case, a pheromone we're really defining in this case as something that's secreted or excreted by the animal. It's a chemical factor or a chemical cue, often volatile or airborne, that triggers a social response in members of the same species. So in the example here on the lower left, this is a typical example of what we would usually classify as a sex pheromone. So either the male, or in this case, the female in this example, produces some sort of a compound that she releases. In this case, in this sort of blue cloud of compounds that are being released, a male is picking that up somewhere downstream or downwind and is navigating towards that female. And so we consider this to be a sex pheromone. The other type of pheromones that are fairly common across insects, especially in dipterans, are what's called aggregation pheromones. These function largely the same. Usually one or sometimes both individuals produce it, but invariably the big difference is that both male and female are attracted to this particular compound or this particular chemistry. So these are really the targets that we are searching for as we start this new project with soybean gall midge. So in general, pheromones are pretty common to most insect orders. Most individuals produce some sort of chemistry that is attractive to other individuals of the same species. There are already huge databases in moths and beetles and in flies. And what's nice is there's a lot of background information that we can start with, so we can cut a lot of corners, I hope, in terms of identifying some of this chemistry. But invariably, it's been fairly successful, especially in things like monitoring. I'll leave it up to others to maybe decide what the best applications are once we can maybe identify a pheromone. But in general, I think this is an example of these images, for example, are codling moth and apple orchards. Generally, you have some sort of rubber septum or some sort of a porous device that you can add a high quantity of pheromone to and apply it to a sticky trap. The idea is that essentially you are calling for these insects to come to your trap. Works very well to monitor and also get a sense of where they are in the field, for example. The really nice thing about pheromones, especially pheromone chemistry, is that it's often specific to one particular insect. So you're not going to be getting a lot of non-target. You're not going to capture a lot of other types of flies or other pest insects, for example. And of course, these are safe for consumption. So they work really well in agricultural systems. And that's really where a lot of my background has come into play. So how are we searching for the soybean gall midge pheromone? We started this project last summer. Essentially, we're combining chemistry and behavior. So we're trying to work with individuals, both male and female in the lab, but especially working with individuals that are in the process of mating. That works much better to collect the chemistry. We're also harvesting a wide variety of insect materials of the different life stages of soybean gall midge. We're also collecting material from the plants as well as microbes like fungus. But for the sake of time, we won't go into that today. We'll just focus on insect-produced pheromones. So most of this work then is using gas chromatography and mass spectrometry. Essentially, we are trying to identify the chemistry that is being released by these insects that will have a behavioral attraction. So just very briefly, in case anyone's interested in how this chemistry process basically works, we're combining two technology, a gas chromatograph and a mass spectrometer. Essentially, the GC is separating the mixture. So if you think about a flower or even a soybean plant, it has a lot of different compounds in it that are all occurring sort of at the same time. The GC uses a column, often many, many meters long, and it allows these compounds to separate essentially in space and time. So this is based on the mass of the compounds, also the charge, and in this particular example here, there are 18 different compounds, for example, that are separated from one particular source or one particular injection. The idea is that you are trying to separate what is a complicated sort of chemistry. The mass spec, on the other hand, you can really think of as a fingerprint. So essentially for each of these peaks in the chromatogram, so in this generic example here, this is CBDV, particular compound of interest for a previous study. The idea is that the mass spec is allowing you to look at these individual compounds as they come off of the GC and look for a fingerprint, or essentially look at these fractions that are occurring to help you identify what that compound is. So by combining these two technologies using the GCMS, our goal is to be collecting samples from the insect to start looking for pheromone components. So there are some complications that come into play that I would just mention, partly because it's going to be helpful as we go through and try to identify some of these. The idea is that there can be several compounds that can produce a pheromone signal. So often in the classical case, or the easiest example, to say, we're going to find one chemical, and that's what we'll use, and that will attract females or males or both. Invariably, there's often major and minor components, and sometimes the ratio or the sort of the relative rate of those two compounds really determines the efficacy or how well it ultimately works in behavior. There's also, of course, enantiomers. So in chemistry, you can think about your two hands. They're largely identical, and yet they're sort of reverse images. Invariably, in pheromone chemistry, only one of these particular chemical configurations will end up being behaviorally active. So often we'll identify many different sort of versions of a chemical compound, and our goal is to narrow it down to which one is the most effective at collecting, monitoring, attracting these animals. The other thing I'd say is, at least right now, we're sort of taking this as an open box experiment. So we're collecting from males, with the idea being it could be a pheromone from the male, collecting from females only, could be from the females. And we're also looking at both and combined and getting an idea both of what is the chemistry being produced, but also ultimately the behavioral attraction. I will say, based on previous work with other insects, I don't know, this is a relatively small picture, but maybe you can see on the right here, the enlarged antenna of the male in this case. It's much more plumose, has much longer seedier hairs. That's usually indicative that the male is searching for the female, and therefore the female is most likely producing the pheromone. But again, it's relatively preliminary, but there's a good chance that that is probably going to be correct. Another aspect is this is the smallest insect I've actually worked with so far. So just size alone makes it difficult to do some confirmation with chemistry, partly because we need a lot of animals to generate a big enough sample that we can be confident in some of the configurations or the chemistry that we're working with. And the other aspect I'd say as we move into some of the behavior aspect of the talk, the next trick is then really concentration. So to really try to find out how do we best optimize once we find the compound, how to best attract the animals. So you can have too little. So the idea is essentially it's too dilute or not strong enough, and essentially the animals won't be attracted. You can also have too much though. So if you overpower or have too strong of a release, that can also lead to sort of lower capture simply because it becomes aversive at higher concentrations. So these are some of the things that we're trying to consider as we're going through and doing some of these tests. Since last summer, based on the field emergence and some of the laboratory emergence, especially coming from Justin's lab, we have been collecting a lot of different samples. In this case, essentially body washes or collecting whole body soaks of individuals, including male and female, across several different extraction protocols, usually related to different solvents. Maybe you can see just based on color here, depending on the type of chemical we're using to extract these compounds, we're getting very different classes and combinations of chemicals. It means going from 1,000 possible compounds, we can start to narrow it down. It looks like, for example, the one on the right, this is DCM, this is dichloromethane. This one seems to so far be the most effective. It seems to work relatively well in some of our preliminary studies with behavior. It has this nice sort of pinkish rose color. It actually looks a lot like both the larvae and the abdomens of the adults. Again, this could be indicative that these colors are related to the chemicals, and especially the pheromones that are being produced by the animals. So again, it's a little too preliminary to tell, but that's the process where we are with going through some of the chemistry. In terms of behavior and trying to really go from candidates where we're going into the lab and trying to identify what could possibly be a pheromone to moving into the behavior and especially the chemical attractiveness, we've started with a relatively simple assay. These are usually called Y-tube olfactometer or a Y-tube choice test. Essentially, we release live adults into the bottom, into the sort of the small funnel area at the bottom of the Y-tube. We put two different smells essentially into each chamber or each side of this Y-tube and essentially look for how the animals respond. So this could be a potential pheromone against a blank control. It could be soybean on one side and a different host plant on the other, or it could be the combination of host plant chemistry with insect pheromone chemistry. Again, we're looking to optimize attraction of the insects here. Again, for the sake of this talk, I'll just say that we're working mostly on pheromones, so insect collected chemistry, but I'm also analyzing plant samples from a variety of different soybean, different parts of the plant, also looking at microbes or fungus that could be associated with some of these infection sites. Again, the goal is to really do classical chemical ecology, trying to identify what compounds can be used to attract this particular insect. So the good news is there's a lot of literature that we're already basing some of our searches on. It's helping us narrow down some potential targets. The raspberry cane midge, for example, in 2009 had a really nice study in the Journal of Chemical Ecology identifying a particular pheromone compound produced by a female that produced strong behavioral attraction. Again, it's within at least the same genus of our particular soybean gall midge. And so again, it gives us some idea of carbon length, some of the different active groups, and it will hopefully help us narrow down our search, going from, again, thousands of compounds down to a handful, which makes it much easier to test. Once discovered, or once we're confident in some of our potential candidates, of course, we'll start in the lab, but ultimately, this will probably be from crude extract. So we'll have, at least for a while, once we can collect enough individuals, initial trials in the laboratory using these pure extractions. But ideally, we may or may not be able to partner with a chemical company to start producing a compound that we could start testing in the field. And just from applications of other insects, some of those applications can be mating disruptions. So that's sort of seen on the left here. Ideally, the idea is you have dispensers that can be releasing quantities of this particular compound, and essentially it confuses whichever individual, male or female, is searching for a partner. And essentially you reduce matings, and therefore you reduce the next generation of offspring. There's also a lot of monitoring that can come from this type of project. We're really hopeful if we can develop a compound that could be used to enhance early detection or detection throughout a field site, for example. These are the types of compounds that we hope to use, and probably in conjunction with some sort of plant host or soybean-based chemistry as well. And of course, there are examples in dipterans. These are tephritid flies, for example, the true fruit flies in the upper right. This is mass capture and removal. So putting these out during a peak aspect, for example, soybean gall midge emergence might be an opportunity to essentially decrease those populations right away. Again, a lot of these things are sort of pretty preliminary at this point. We've really only had a few months to get started on this, but I'm really looking forward to joining this wide group of researchers, and again, to take more of a chemical ecology approach to look for compounds of interest to help us better understand the navigation of how these insects are finding each other in this case. So with that, I'd really like to thank you for your time and attention. I tried to keep it short to maybe put us back on time here. I'd like to thank certainly my graduate student, Sebastian Kailman, who has started with me now. He's a PhD candidate, and also the undergrad, Sam Mackley, who's a junior pre-med student who's also helping now with some of the behavioral trials. Again, hopefully by next year, we'll have a lot of data to go along with the presentation. But for now, thank you for your time and your attention. Happy to answer any questions.
Well, we had one question so far. How realistic would it be to develop pheromone traps from insects that release low concentrations of pheromone? Would you synthesize them?
Yeah, so I have to say, it's always a complex process going from identifying the pheromone to getting something out into the field. I think we have a good head start, partly because we have a lot of other examples of other similar flies. So we have some background on what has worked in other field trials, for example. But I do think it is likely that we will be able to identify a compound that will be effective in terms of trap capture.
Okay.
Okay, thank you. I think we'll move on to our next topic now. We're going to have a couple speakers from University of Minnesota. Sarah Lesick and Gloria Milotto, and they'll be talking to us about biocontrol.
Okay, so good afternoon. My name is Gloria, and today Sarah and I will share a little bit of our work on biological control of soybean gall midge that we are developing here at the University of Minnesota as master's students. So first of all, what is biological control? So biocontrol is the use of living organisms, so known as natural enemies, to suppress pest population in field conditions. So these natural enemies can be predators, parasitoids, and pathogens. But today we are going to focus on predators and parasitoids as putative agents of SGM biocontrol. So all work that will be presented today by us was done in 2 fields in 1 farm in Luverne, Southwest Minnesota. So, and I will start with the research that we performed to find the parasitoids that attack soybean gall midge. So a parasitoid is a wasp that lives in close association with its host. So they often lay their eggs inside the host, and then its larva will eclode and feed on the host, and eventually the host will die. So to discover which parasitoid attacks SGM in field conditions, we collected injured soybean stems in the field, and we brought back to the lab. I cut the stems I have here in this photo and wrapped the tip with parafilm and placed them into emergence cages that we designed here that were maintained in the lab. So I checked the cages every day and collected the parasitoids and the soybean gall midge adults that had emerged. And as a result of these emergence cages, we had 17 parasitoids that came out. And with the help of a taxonomist, they were identified as the genus Cynopias, that is a genus known to parasitize gall midges. However, after checking DNA from these wasps, we discovered that the DNA and the morphology didn't match any known species. So which means that we have a new species of parasitoid that the description is on the right now. Okay, so now that we know which parasitoid attacks soybean gomage and we have its DNA, we are going to evaluate parasitism rates in field conditions by molecular methods. So for this, we collected injured soybean stems in the field to dissect the stems, count and collect the SGM larvae that we found, and preserve them in alcohol and in the freezer until extracting the soybean gomage larva DNA individually to perform real-time PCR to see if we could detect parasitoid DNA in soybean gomage larvae that we collected in the field. So qPCR is a really sensitive method of detection of DNA. So in 2021, I screened almost 3,000 soybean gomage larvae. In 2022, I screened 4,500 larvae. So after detecting parasitoid DNA in some of larvae samples, we confirmed that the larvae that were parasitized was indeed soybean gomage by DNA barcoding some of them. So here I will show you the results of this molecular screening. I have data of sampling in the x-axis and proportion parasitized in the y-axis. And the data is separated between field edges in the left and interior in the right. So in 2021, here in red, we observed parasitism only on field edges and at the beginning and at the end of the season. And in 2022, that I have here in blue, last year, we were able to detect parasitism in the interior of the field, which can maybe mean that the parasitoids are moving towards the interior of the field. And we also observed higher parasitism rates at the end of the season. So during these 2 years of research investigating parasitism of soybean gomage, we discovered a new species of parasitoids that naturally attacks soybean gomage here in Minnesota. And these natural parasitism rates are higher at the beginning and the end of the season. We don't know yet the reason for this, but it can be because of the biology of the parasitoid. However, this work is an initial step, and more research is needed to investigate how to keep these insects active in the field and also how to increase their density, maybe by reducing the use of insecticides or planting flowers that may attract them, but there's a lot of space for research here. And as a future work, we will expand molecular screening to other states. So our plan is to extend to Iowa, Nebraska, and South Dakota.
All right, hello everyone. My name is Sarah, like Gloria introduced me, and I'll be talking to you about predators. So first, Gloria sampled soybean fields for predators in 2021 in order to better understand which predators are found among SGM's environment. So to do that, she used pitfall traps shown here on the left, sweep nets, and also plant inspections. And she found all sorts of different predators, including spiders, lady beetles, lacewings, minute pirate bugs, crickets, and various ground beetles. And so here's a graph. We have all of the numbers of the predators found in these Minnesota soybean fields in 2021. We have the number on the y-axis, and on the x-axis, we have our predators. And if you notice on the y-axis, there's a break between 250 and 4,250, and that's because we had one predator that was the most prevalent overall, and that was this ground beetle, Pterostichus melanarius, with over 4,000 individuals collected. So this beetle is a common ground beetle and a key predator in soybean fields, and therefore a potential biological control agent. Gloria also documented that this beetle will feed on SGM. So this leads to the question, do these beetles have a preference for SGM over other prey that are found in the field? So we decided to compare it to another common prey found in soybean fields, the soybean aphid. And this beetle is also known to feed on soybean aphid. So we set up a prey choice test. And to collect all of our different insects, we used pitfall traps for the beetles. We dissected stems, infested stems for the larvae, and then we used a lab colony to collect the aphids. So this prey choice test was to test if this beetle prefers SGM over SBA or soybean aphid. So each beetle was given one of 3 treatments of varying ratios of SGM to soybean aphid. These are the different ratios you can see here on the screen. We have 3:9, 6:6, and 9:3. And the numbers of the prey consumed were observed over time at 3 hours. So here we have a graph where on the x-axis we have the ratio offered, and on the y-axis we have the ratio consumed. And any data point that will fall in the orange shaded area will show SGM preference, and anything falling in the green area will show aphid preference. So if we put our data points on this figure, this is what it ends up looking like. So you can see the majority of the data points fall in the orange shaded area, and after doing some statistical analyses, we see that there was a statistical difference where there was a preference for SGM. Moving on to our next study, which is our field predation study. The goal of this study is to determine predation rates in the field using molecular tools to detect SGM DNA within the beetle gut. So basically we're trying to see, do they actually eat them in the field, and how many of them in the fields actually get eaten from the beetles. So those same beetles that Gloria collected to determine the predators at the beginning of this talk were saved, and then now we're going back and processing them. So they were frozen, and now we're actually diving into them and dissecting their guts. So we dissected, as you can see here, the guts from the beetles and used molecular detection of SGM DNA in the gut. And here we have our initial results. So this is just from one day. On July 28th, 2021, we see that out of 80 beetles collected, 6 came back positive for SGM DNA detection, which is 7.5% of beetles that were collected actually ate SGM. So it's important to note that this is an underestimate of the field predation, since there's only so much time before SGM is digested through the beetle gut. So there's a narrow window of time where we can actually detect SGM in the gut. So moving on to future work, the plan is to repeat the prey choice test under field conditions, to continue processing field samples from multiple dates and multiple years, and then also to determine the length of time the DNA remains detectable in the beetle guts. So in summary, the predator that we found in the soybean fields that was most prevalent was this beetle, and it can be found in multiple soybean fields. It is also known to feed on SGM in both the lab and the field setting, and it prefers SGM over soybean aphid. And this is all very interesting, but what does it tell us? So knowing this beetle has a preference for SGM, it looks promising as a biological control agent. And it would be beneficial for growers to try to implement conservation biological control. And 2 ways to encourage these beetles is by applying insecticides only when needed. So this minimizes the killing and harming of beneficial insects like this beetle, and also reducing tilling. And this will create hiding spots for the beetle with the leftover debris, and also will minimize ecosystem disturbance within the fields. Thank you to everyone who's listed on here. And now Gloria and I will take questions.
Okay, there was one question you sort of already addressed, but what impact do you think insecticide seed treatments or foliar applications of insecticides could have on parasitoids and potential predators of SGM? Obviously, the parasitoid is newly identified or newly described, so you don't have any research, but are you aware of effect of foliar insecticides or seed treatments on carabids?
So the thing is, like, the field that we have our research going on, they do seed treatment.
Okay.
So with insecticides, fungicides, but they did not apply any foliar insecticide. So even though they do seed treatment, we see SGM larvae and we observe parasitism and predation.
But I think I'm Bob Cook with the University of Minnesota. I think one thing we know from other systems is that a lot of these natural enemies are sometimes more sensitive to the insecticides than the pests even, you know, so we know these parasitic wasps are oftentimes more sensitive to these foliar applications of insecticides. I think some of the work has shown that seed treatments can have adverse impacts on ground beetles, right? So I think we need to be thinking about how we're using our insecticides to try to promote this additional tool for managing the pest, because we know some of the other tools alone aren't very effective. So I think after seeing all these different presentations, we don't have any one silver bullet yet, you know, so I think we're gonna have to think about how we can integrate these all these different tactics into one management program.
Okay, we had several other questions. Do cover crops help increase predator populations?
We don't have any data on that for this system, but you know, some ecological theory suggests that having that increased plant diversity there can, in some cases, help the beneficial insects, be it predators or parasitoids. But again, we just haven't had a chance to test that for this particular system yet with the soybean gall midge and the natural enemies we're looking at.
Okay, and here's the $1 million question. Will we see a similar pattern as with soybean aphid, where eventually natural predators will keep populations low? We can only hope, right?
Yeah, let's hope so.
Has anybody looked at entomopathogenic fungi or nematodes targeting the larvae or pupae in the soil?
As far as I know, no one has done any of that, unless, Bob, if you know of anyone from Nebraska working on that, but I don't think that's been done for this particular pest. Some work has been done along those lines for other gall midges. So I think as we, you know, get more time, we can, you know, kind of expand the horizons for the biocontrol work and start looking at some of these other different kinds of natural enemies.
There's a question about the carabids. I just interject, part of the answer is they only feed on, at least this carabid only feeds on other insects or other organisms, but may the predator beetle present negative impacts to other crops or on beneficial insects? You know, there are some carabids like the seed corn beetle that'll feed on plants as well as insects.
Yeah, generally speaking, I think this particular species has been considered beneficial and a lot of the others, you know, as predators of other, you know, pest insects, but also some of them will eat weed seeds and they can help reduce the seed bank for some of the weeds. You know, it's possible that they might be feeding on maybe some other natural enemies out there, but I think the research is generally considering this ground beetle that Sarah talked about as a beneficial.
And the question is, what stages of the soybean gall midge does the beetle feed on?
Sarah, do you want to?
We have only tested the soybean larvae, I believe. You haven't tried an adult right now. Yeah, so we don't know the pupae, anything about that. We haven't tried feeding them that, but they do eat the larvae.
And these are mostly on the soil surface, so they may not interact with the adults that frequently, but anyway.
Yeah, I mean, another aspect that we don't know is, are these beetles, do they have the ability to dig down into the soil? To find the soybean gall midge larvae or pupae there, and the larvae of this predatory beetle occurs in the soil.
Yeah.
And they're known to be predators as well, but we haven't had a chance to evaluate them as predators of soybean gall midge yet.
Thank
Okay.
all the students for their talks today. Great job. It's a tough environment to get presentations in. You all did excellent. So thank you.
Yep, it was very good, very exciting, particularly once we got out of the insecticide. Insecticides have been kind of disheartening when we're working with them, but some of the hilling and then the pheromones, host plant resistance, biocontrol is pretty exciting.